So first of all, is there any evidence that aggrecan and that astrocytes affect integrin signaling? We can look at that through photoadhesion kinase labeling. You can see aggrecan decreases phospho-FAC. That indicates inactivation of integrins. Astrocytes, if you look at Schwann cells in contact with astrocytes, also express lower levels of photoadhesion kinase. So we think what's happened is that the aggrecan has inactivated integrins. What if we force them into an active state? We can do that by treating the cells in a variety of ways, with manganese, which is rather crude, or with an integrin activating antibody, such as TS216. Well, if you force integrin activation, you find that the Schwann cells can now migrate perfectly well, even on an inhibitory aggrecan surface. So here's a control, here's cells migrating in the presence of manganese, here's some quantification, and cells migrating over astrocytes. So activation of integrins is a mechanism for overcoming inhibition both by aggrecan and by astrocytes. And we think that one of the ways in which aggrecan prevents migration is through inactivating those integrins. A second mechanism, and I'm afraid there's three of these, which I'm going to talk about too, is Fs and ephrins. Now, there's a wide literature on cell mixing and cell separation during development based on the interactions of Fs and ephrins. We did a screen to see if any of the ephrins that might be produced by the nervous system or by astrocytes inhibit Schwann cell migration. And this is the screen. And the one that was the most inhibitory is ephrin A5, and that's a quantification here of the inhibition of migration by ephrin A5. And that slide there, that picture there, demonstrates that astrocytes produce ephrin A5, this inhibitory molecule. So again, ephrin A5 is produced by astrocytes. The receptor, FA4, is produced by Schwann cells, and therefore you can dream up a mechanism by which the inhibitory molecule produced by astrocytes affects the receptor on Schwann cells, which then affects Schwann cell migration. And if the Schwann cell migration is on laminin, as I'm going to be showing you, then it's likely to have acted through an effect on integrins. So first of all, does blocking ephrin A5 do anything? You can do this by putting on an excess of the receptor FA4 as an FA4-FC. Here's a control confrontation culture. Here's one which has been treated to block ephrin A5, FA4 interaction. And you can see that there's increased integration of the Schwann cell into the astrocyte boundary. Now, how does this work? The signaling mechanism downstream of Fs and ephrins depends, in many cases, on some signaling molecules called VAVs. Now, the VAVs are expressed in Schwann cells, and what we find is that VAV phosphorylation is increased where Schwann cells touch astrocytes. So there's an astrocyte, there's two Schwann cells in contact. I think you can see that the blue is more intense than those Schwann cells in contact with the astrocytes. In other words, Schwann cell astrocyte contact increases VAV activation in the Schwann cells. If we knock down VAV in the Schwann cells, again, we do have a positive effect on Schwann cell migration on an astrocyte surface. So again, it looks as if ephrin A5 rather, is an inhibitory molecule produced by astrocytes that signals at least partly through this mechanism, VAV2. What does it do? I've taken out the slides for this because we're rather short of time, but basically we have evidence, just as I showed you before, that it downregulates the phosphorylated form of photoadhesion kinase and inactivates integrins, and therefore decreases cell migration through an effect through integrins. So the final mechanism, which again I'm not going to talk about, is adhesion. So if cells adhere too strongly to one another, they can't migrate. There's a bell-shaped curve. No adhesion, cells don't migrate. A reasonable level of adhesion, cells can migrate over one another. Excessive adhesion, the cells again get stuck down and are unable to migrate. There's adhesion between Schwann cells and astrocytes that is mediated by N-cadherin. And again, if you block N-cadherin interactions, you can increase Schwann cell migration over astrocytes. So this at the moment is all in vitro studies. In vivo studies are being done in a number of laboratories, including Mary Buggy's in Miami, Mike Failey will be telling you about some results they're going to be getting, and we're also doing some in Cambridge. So just to summarize, there are various mechanisms, 3 mechanisms that we now know about, through which astrocytes block Schwann cell integration into spinal cord tissue. First of all, the astrocytes produce inhibitory ephrins, of which the most inhibitory is ephren A5. Schwann cells, astrocytes rather, produce the inhibitory protoglycan aggrecan, also some other inhibitory protoglycans, and these inhibit Schwann cell migration, and that can be largely neutralized by chondroitinase. Both these mechanisms act at least partly through inactivation of integrins, and another mechanism of overcoming inhibition is to force the integrins into an active state. The strong adhesion mediated by NCAT is a third mechanism, which again slows Schwann cell migration on astrocytes. So our conclusion is that there are several mechanisms through which the nervous system prevents transplanted cells integrating and migrating. These mechanisms are likely to be general, not just to Schwann cells, but to other sorts of transplanted cells. But at any rate, these results point to a number of ways in which you can potentially intervene to alter your transplanted cells or to alter the host milieu in order to improve the integration of transplanted cells. I'll finish just by acknowledging the people who did the work. The Schwann cell migration work has been done by 2 PhD students in the lab, Farhad Afshari and Pippa Warren, and the extracellular matrix investigations were done by Jessica Koch, and there are various funding organizations. Thank you very much for your attention.